Hi, I'm Kathy Bryson, and I'm writing tutor at the Learning Support Commons in Seminole. And I'm here today to read banned books. I'm going to read The Immortal Life of Henrietta Lacks. This is a great book, and it's kind of ironic that it's being challenged in Tallahassee, even as we speak, in October 2015. Because this book is it's interesting for two reasons. One, it's not fiction. A lot of times fiction gets the bad rap. This is nonfiction, and it's about research and the problems that have plagued research, where that fine line is between needing to do research and being respectful of the human beings who are part and parcel of it. So I think it's a lovely example of the problems that we have with challenged books. We want to be respectful of people's beliefs, and also only put out not give people books that are inappropriate. Children don't necessarily need adult books. It's a tough job for the librarians, because at the same time they want to promote freedom of press, right to read, and critical thinking. So I bring you today The Immortal Life of Henrietta Lacks. The prologue. There's a photo on my wall of a woman I've never met. Its left corner torn and patched together with tape. She looks straight in the camera and smiles, hands on hips, dress suit neatly pressed, lips painted deep red. It's the late 1940s, and she hasn't yet reached the age of 30. Her light brown skin is smooth, her eyes still young and playful, oblivious to the tumor growing inside her, a tumor that would leave her 5 children motherless and change the future of medicine. Beneath the photo, a caption says her name is Henrietta Lacks, Helen Lane or Helen Larson. No one knows who took that picture, but it's appeared hundreds of times in magazines and science textbooks, on blogs and laboratory walls. She's usually identified as Helen Lane, but she has no name. Often she has no name at all. She's simply called HeLa, the code given to the world's first immortal human cells. Her cells cut from her cervix just months before she died. Her real name is Henrietta Lacks. I've spent months staring at that photo, wondering what kind of life she led, what happened to her children, and what she'd think about the cells from her cervix living on forever, bought, sold, packaged, and shipped by the trillions to laboratories around the world. I've tried to imagine how she'd feel, knowing that her cells went up in the first space missions in zero gravity, or that they helped with some of the most important advances in medicine: the polio vaccine, chemotherapy, cloning, gene manipulation, in vitro fertilization. I'm pretty sure that she, like most of us, would be shocked to hear that there are trillions more of her cells growing in laboratories now than there ever were in her body. There's no way of knowing exactly how many of Henrietta's cells are alive today. One scientist estimates that if you could pile all HeLa cells ever grown onto a scale, they'd weigh more than 50 million metric tons, an inconceivable number given that an individual cell weighs almost nothing. Another scientist calculated that if you could lay all HeLa cells ever grown end to end, they would wrap around the Earth at least 3 times, spanning more than 350 million feet. In her prime, Henrietta herself stood only a bit over 5 feet tall. I first learned about HeLa cells and the woman behind them in 1988, 37 years after her death, when I was 16 and sitting in a community college biology class. My instructor, Donald DeFleur, a gnomish balding man, paced at the front of the lecture hall and flipped on an overhead projector. He pointed to 2 diagrams that appeared on the wall behind him. They were schematics of the cell reproduction cycle, but to me they looked just like a neon-colored mess of arrows, squares, and circles with words I didn't understand, like MPF triggering a chain reaction of protein activations. I was a kid who failed freshman year at the regular public high school because she never showed up. I transferred to an alternative school that offered dream student studies instead of biology, so I was taking Dethler's class for high school credit, which meant that I was sitting in a college lecture hall at 16 with words like mitosis and kinase inhibitors flying around. I was completely lost. "Do we have to memorize everything on those diagrams?" one student yelled. "Yes," Dethler said. "We had to memorize the diagrams, and yes, they'd be on the test. But that didn't matter right then. What he wanted us to understand was that cells are amazing things. There are about 100 trillion of them in our body, each so small that several thousand could fit on the period at the end of this sentence. They make up all of our tissue, muscle, bones, blood, which in turn makes our organs. Under the microscope, a cell looks a lot like a fried egg. It has a white, the cytoplasm, that's full of water and proteins to keep it fed, and a yolk, the nucleus, that holds all the genetic information that makes you you. The cytoplasm buzzes like a New York City street. It's crammed full of molecules and vessels endlessly shuttling enzymes and sugars from one part of the cell to another, pumping water, nutrients, and oxygen in and out of the cell. All the while, little cytoplasmic factories work 24/7, cranking out sugars, fats, proteins, and energy to keep the whole thing running and feed the nucleus. The nucleus is the brains of the operation. Inside every nucleus within each cell of your body, there's an identical copy of your entire genome. That genome tells cells when to grow and divide and make sure they do their jobs, whether that's controlling your heartbeat or helping your brain understand the words on this page. Devler paced in front of the classroom, telling us how mitosis, the process of cell division, makes it possible for embryos to grow into babies and for our bodies to create new cells for healing wounds or replenishing blood we've lost. It was beautiful, he said, like a perfectly choreographed dance. All it takes is one small mistake anywhere in the division process for cells to start growing out of control, he told us. Just one enzyme firing, just one wrong protein activation, and you could have cancer. Mitosis goes haywire, which is how it spreads.